Hi everyone! I'm very excited to introduce you to the topic of plant central metabolism. Before we dive into the topic, I want you to take a moment to appreciate the wide range of molecules that plants make for us. Many of these compounds or metabolites here are considered as specialized metabolites, which plants use to attract pollinators or fight against a pathogen. It's fascinating to know what's going on with these metabolites inside plant cells and what are the chemical pathways that transform these metabolites. Before getting there, we need to have a good understanding of plant central carbon metabolism that is essential for plant growth and development, because central metabolism provides the building blocks for making all these specialized metabolites. In this video, I will briefly review the Kelvin-Benson cycle and the photorespiration that you have learned. Then I'm going to introduce starch and sucrose synthesis, glycolysis, citric acid cycle, or the TCA cycle, and oxidative phosphorylation. In the next video, I will cover the oxidative pentose phosphate pathway and the lipid metabolism. Although I'm introducing these metabolic pathways individually, I want you to think about metabolism as a giant subway system. Metabolites are being converted or shuttled across membranes by various enzymes. These different pathways are highly interconnected. And to understand the functional state of metabolism, we do need to know how fast these molecules are actually moving through a pathway versus another. And that's what metabolic flux analysis is about. It can give us a map with traffic information of different pathways, which is the cutting-edge research in metabolism. Okay, let's start with the review of the Kelvin-Benson cycle and the photorespiration. During photosynthesis, CO2 enters into Kelvin-Benson cycle. For every 3 rounds of the cycle, 3 CO2 molecules enter the cycle, providing 3 new atoms of fixed carbon. Then 6 triose phosphate molecules are made. 5 of them are recycled to regenerate 3 molecules of the RUBP. One of the triose phosphate is gonna come out of the cycle. We will talk about it in a minute. Moving on to photorespiration. It is the pathway that detoxifies 2-phosphoglycolate, a product of the oxygenase reaction of Rubisco enzyme. This 2-phosphoglycolate is converted into glycine by a series of reactions. Then glycine is going to serine with the loss of 1 CO2 molecule, which wastes about 1/4 of the carbon fixed by photosynthesis. This serine will eventually become 3-phosphoglycerate, re-entering the Kelvin cycle. These 2 pathways carry the largest fluxes of carbon in leaf metabolism. Now we have some triose phosphates coming out of the Kelvin cycle. Where are they going? Some of them are gonna produce sucrose in the cytosol that will then be exported to the rest of the plant. Some of them are gonna store as starch in the chloroplast so that during the night, when photosynthesis is not possible, the stored starch can be broken down and used as carbon source. First, let's look at how starch is made in chloroplasts. Starch is essentially a chain of glucose molecules. So how do plant cells stitch a bunch of glucose together to form long chains of starch? First, 2 triose phosphates will end up in 1 glucose 1-phosphate. The problem is glucose 1-phosphate is not reactive enough to attach to another glucose. So we have to activate the glucose 1-phosphate into a high-energy form, which is ADP glucose. The key enzyme here takes a glucose 1-phosphate and reacts it with ATP so that it sticks an ADP onto the glucose. Once the activated UDP glucose is formed, it can be attached onto a growing starch chain. Sometimes the chain is straight, sometimes it's branchy. Oftentimes, one starch molecule could be made up of 100,000 glucose molecules. They are packed into a compact shape which takes less space and good for storage. You can actually see the starch granules in chloroplasts under high-resolution light microscope. They are insoluble in water, which means not very reactive. Another good reason for storage. Moving on to sucrose synthesis. In this case, Chile's phosphate is being exported from the chloroplast into the cytosol, going through similar reactions as starch synthesis, until we have glucose 1-phosphate. For the same reasons, we want to first activate glucose 1-phosphate by sticking an UTP on it. In starch, it was adding ATP, but the purpose is the same, making glucose more reactive. Once we have UDP glucose, it can polymerize with fructose 6-phosphate to make sucrose phosphate, and then the phosphate has to be hydrolyzed from it to form sucrose. Note that sucrose is a disaccharide which has 2 hexyl units, therefore 12 carbon atoms in total. There are some similarities between starch and sucrose synthesis. The differences are starch synthesis occurs in the chloroplast and use ATP to activate glucose, while sucrose synthesis occurs in the cytosol and use UTP to activate glucose. Another difference is that rather than making a long-chain polymer of glucose in starch, you are making just a disaccharide for sucrose. Plant central metabolism is very dynamic. The metabolic traffic will be completely different between photosynthetic tissues and non-photosynthetic tissues like roots. Day traffic would also be different from night traffic. During the day, leaf cells use triose phosphates to make starch in the chloroplast. At night, the stored starch is broken down into maltose and glucose, which eventually contribute to sucrose synthesis in the cytosol. The levels of chloroplast starch are maximum during the day and minimum at the night. We just talked about some of the triose phosphates in chloroplast are flowing to cytosol to make sucrose. In most plants, sucrose is the main transport form of fixed carbon because it's less reactive than glucose or triose phosphate. Sucrose are transported from leaves to the rest of the plants, such as growing roots, stems, and tubers. Well, the next question is, how do these non-photosynthetic tissues or cells release the stored energy from sucrose to fuel growth? Now we are transitioning to a big topic of respiration, a universal process in all living cells. Let me give you an overview of the 4 pathways in respiration. Glycolysis is the first step of respiration. It begins with glucose. So to be able to use sucrose, plants need to convert sucrose back to glucose. Glycolysis involves reactions in both the cytosol and plastids. Its main role is to split the 6-carbon glucose into organic acids and generate a small amount of energy in the form of ATP and NADH. Moving down to mitochondria, organic acids from glycolysis are going to enter the TCA cycle, where you have CO2, NADH, and FADH2 coming out of it. The last step of respiration is oxidative phosphorylation. We are giving all the electrons from NADH and FADH2 to oxygen and gather all the energy to make ATP. This also occurs in mitochondria. Oxidative pentose phosphate pathway is an alternative way to use the hexose phosphate. It occurs both in the cytosol and the plastids. It oxidizes a hexose phosphate to a pentose phosphate, So one carbon is lost as CO2 in this process, and the reducing power NADPH is generated. Now let's zoom in a little bit. Glycolysis is a series of reactions that transform the 6-carbon glucose into 3-carbon pyruvate and 4-carbon malate. The early steps of glycolysis, from glucose to triose phosphate, requires an investment of 2 ATP. This is the source of the phosphate groups in these phosphorylation reactions. The fructose 1,6-biphosphate splits into 2 different 3-carbon triose phosphates. One of these is quickly converted to the other, which is glyceraldehyde 3-phosphate. From now on, everything is duplicated because there are 2 glyceraldehyde 3-phosphate for every glucose coming down the pathway. The later steps of glycolysis from triose phosphate to pyruvate is the payoff phase, where you gain 4 ATP and 2 NADH. Let's think about an extreme scenario. When plants are flooded with water, then oxygen becomes limiting. Glycolysis may be the only pathway available to generate ATP, and this is where the fermentation pathway comes in. If pyruvate cannot enter mitochondria for the TCA cycle, it is converted into either ethanol or lactate. Ethanol and lactate themselves are not so good for plants, but these processes regenerate NAD+ to keep glycolysis going. Note that in this step we need NAD+. So as long as glycolysis is going, we can still generate a little ATP, which is needed for cells under low oxygen. Under normal conditions, pyruvate generated in glycolysis are going to move into mitochondrial matrix. Pyruvate is converted into acetyl coenzyme A, or acetyl CoA in short, before entering the TCA cycle. Acetyl CoA is a fascinating molecule that we will see again in lipid metabolism. For now, you just need to know it's a high-energy molecule with 2 carbon atoms. From pyruvate to acetyl CoA, 1 CO2 is lost and 1 NADH is generated. Then acetyl CoA enters the cycle by combining with a 4-carbon molecule, oxaloacetate, to form the 6-carbon molecule citrate, then to isocitrate. The 6-carbon isocitrate is going to lose 1 CO2 to become a 5-carbon alpha-ketoglutarate, also known as 2-oxoglutarate, and then continue to lose 1 more CO2 to become a 4-carbon succinyl-CoA, then succinate, fumarate, malate, and ending with oxaloacetate, which completes the whole cycle. Along the way, each citrate molecule will produce 1 ATP, 1 FADH2, and 3 NADH. You may say the TCA cycle doesn't produce much ATP. That's true, but it makes NADH and FADH2. The energy from these electron carriers will be used in the oxidative phosphorylation to produce the bulk of the ATP payout, which we will talk about in a minute. Another important role of the TCA cycle is to provide the carbon backbone for amino acids synthesis. For example, glutamate is derived from alpha-ketoglutarate, and aspartate uses the carbon backbone from oxaloacetate. Finally, we have oxidative phosphorylation. It involves a series of protein complexes embedded in the inner membrane of mitochondria, also known as the electron transport chain and ATP synthase. Electrons from NADH, FADH2, and NADPH are going to deposit their electrons to the electron transport chain. The final electron acceptor in this system is oxygen. That's why plants also need oxygen like us. This process of transferring electrons drives the pumping of protons, leading to the proton gradient, with a higher concentration of proton in the intermembrane space and a lower concentration in the matrix. The flow of proton through the ATP synthase makes it to spin, which catalyzes the formation of ATP. This is how plants get the majority of ATP out of glucose. In this video, we talked about how plants make starch for storage and the sucrose for transport. We also talked about how plants release the energy from sugars by glycolysis, the TCA cycle, and the oxidative phosphorylation. We will continue with oxidative pentose phosphate pathway and lipid metabolism next time. See you then.